Hello, everyone, and welcome to the VMC Animal Health Education Series. My name is Lauren Kraft, and I'll be your host this evening as we hear from leaders in veterinary medicine as they share their expertise in areas ranging from relevant health information for your beloved pets to ways that we're advancing clinical research that will serve dogs, cats, and people for generations to come. During the presentation, you can use the Q&A function in Zoom to ask questions. We ask that you use the Q&A button instead of the raised hand button, as it helps us better track the questions as they come in. We also ask for pre-submitted questions, and we'll do our best to cover the themes that came up most frequently during the final portion of the program. Please note that if your question for tonight's presentation relates to your pet's specific medical care, it is best to call the VMC directly or to have your primary care veterinarian contact our team for a consultation, so that we can best serve you and your family. With that, I'd like to introduce our speaker for tonight, Dr. Stephen Friedenberg. Dr. Friedenberg is an assistant professor in the College of Veterinary Medicine at the University of Minnesota, a researcher in the Canine Genetics Lab, and a member of the Emergency and Clinical Care Service at the VMC. Dr. Friedenberg's primary interests include autoimmune disorders such as Addison's disease and IMHA. His research focuses on understanding the genetic and immunologic mechanisms that cause these diseases in dogs. We're grateful to have Dr. Friedenberg with us tonight to discuss complex genetic traits and related research that we're pursuing to help improve patient care and outcomes. With that, I'd like to welcome Dr. Friedenberg.
Good evening, everyone, and thank you so much for coming to this talk tonight. I really enjoy being here, and also thank you, Lauren, for the really nice introduction.
Your deck looks good.
Deck looks good. Okay, great. I just want to make sure everything was in the right place before we get started. So again, thank you everyone for coming. I'm really looking forward to being here, to talking to you guys a little bit about complex trait genetics, which is the kind of research that I do, and then to spending some time at the end answering any questions that you might have about things that come up in the talk or things that have been kind of submitted in advance. So all of that said, kind of let's go ahead and get started with the presentation. So my motivation for this talk, so obviously I'm a geneticist, and that's sort of what I spend the vast majority of my time doing. And as Lauren mentioned, I study diseases like Addison's disease, an autoimmune disease called IMHA. And these diseases are complex traits. So it takes kind of we'll get into a little bit later in the talk exactly what a complex trait is. But oftentimes I get asked by people, by breeders, can you potentially work on some disease that I'm interested in a breed that I'm interested in? Or people will oftentimes ask me, why is it so hard to find the mutation for such and such a disease? We've already found the mutation for this disease. Why is it so hard to find the mutation for that disease? And a lot of the reason why mutations can be really, really hard to identify is because many of the traits that cause very, very common medical problems in dogs, cats, humans, whatever, are complex traits, and they take a lot more work to dissect and understand. And so I thought I'd talk to you guys this evening about what a complex trait is, how it differs from easier to understand genetic traits, and then also to explain what researchers like me, as well as many others in the canine genetics community, are doing to try to understand exactly how these complex traits work. So with all of that said, I'm going to give you a little bit of an overview of the presentation that we're going to go through tonight. So I'm going to start out by going through some definitions that I think are just important to understand as we think about complex traits. For example, what are things like heritability? What is a gene? How do genes make proteins? We'll then start off by talking about Mendelian traits and what is a Mendelian trait, and explain how those are the most common diseases that have been identified genetically in dogs these days, to explain how there's even some complexity around Mendelian traits. And then we'll switch gears and talk about complex traits. What are complex traits? And then lastly, we'll end the presentation by trying to understand this problem, which I'll explain as we go through our talk, around something called missing heritability, which is essentially some of the challenges that we face today in understanding these complex traits. So we're going to start off with some definitions and then go from there. Okay, so the first thing which I want to define for everyone here is this notion of something called heritability. And heritability is a concept that geneticists talk about frequently, and it essentially explains in a quantitative way how much of a particular disease can we explain by genetics, right? So I kind of get to that nature versus nurture hypothesis that I'm sure many of us are familiar with just from reading newspapers, magazine articles, et cetera. And I thought this figure that I have here on the right, it actually comes from 23andMe. And this is something that's been defined in human genetics, where there's a lot more researchers focusing on problems. And heritability has been quantified for a number of different common human diseases. And these diseases are shown here at the bottom, where you see diseases like bladder cancer, lung cancer, colorectal cancer, all up to diseases here toward the right, like celiac disease, for example. And heritability is the number here on the up and down on the y-axis that ranges from a number of 0 to 1. So a disease that has a heritability of 1, we can say that genes would be entirely responsible for the development of that disease. Whereas a disease that has a heritability of 0, we would say that genes have a 0 role or no role in the development of the disease. And what you can see in this graph is that different diseases have lots of different heritability. So how much do genetics actually contribute to the development of the disease? So take a disease here for example, in humans in the middle, something like coronary artery disease. So that's a common disease. We know it leads to heart attacks. And to some extent, right, we can explain some percentage of the cases of humans who develop coronary artery disease by genetics. But there's a whole component of the risk of developing that disease that we can't explain by genetics. And the reason why I wanted to kind of bring this concept of heritability up in the beginning is because many of the diseases that people think about or that people want to understand, hey, how do I test my dog for this disease? Or how do I know if my dog's going to develop this disease? Is that they may not be completely heritable, meaning there's no way we're ever going to be able to say with 100% certainty with only genetics, will my dog get this disease? So that's really the first concept that I just want to bring out and highlight up front, because it limits what we can really do with genetics and understanding disease risk. The second concept, which I just want to bring up here, is something called a gene. So what is a gene, right? We throw this word gene around a lot when we're thinking about genetics and mutations and things like that. So I think most of us know that DNA is the basic building block that lives inside of all cells that sort of creates our genetic blueprint, is the way that we oftentimes think about it. And I've shown a molecule of DNA here on the right here, where you see DNA, and DNA is shaped here like a double helix. And DNA, when you sort of look at the guts of it, is broken up into the most important parts, which are these letters, which are called A's, T's, G's, and C's. As geneticists, we call them bases. And in the dog genome, for example, in the human genome, there's about 3 billion of these bases in every single copy of DNA. Now, a gene is just a fragment or a small piece of the DNA that lives inside of our cells. But the reason why we have it as call it a discrete unit is because one gene encodes or sort of provides the instructions for a particular protein that goes on to carry out a function in the body. So that's what a gene is. It's just a segment or a fragment of DNA that goes on to encode a particular protein or some other gene product. And so why is this important is because we think of mutations and things that cause diseases as occurring within the context of a gene. So it's really, really important to understand that a gene is just a stretch of DNA composed of these letters and bases. Okay, next basic concept that I want us to get to is to understand a little bit about this thing we call the central dogma of molecular biology. And that's to understand a little bit about how DNA turns itself or really provides the instructions for encoding proteins, which then go on to do the stuff of life, right? To make our cells function, to make our heart beat, to make our liver work, to make us see, to make us hear, to make us think and understand. It's really proteins that are doing that work, but it comes from the underlying DNA. So the central dogma of molecular biology explains how DNA gets us to proteins. And essentially it starts here on the left, where you have this big molecule of DNA. A certain fragment of that DNA, typically a gene, it undergoes a process where it's transcribed into this messenger molecule, which we call RNA. RNA then moves into a different portion of the cell where the RNA undergoes something called translation, where it turns that RNA into an amino acid chain. So amino acids are the building blocks of proteins. And then once those amino acids fold in a particular way, they actually form a protein that goes on to do the stuff of life, as I mentioned before. So this is how DNA provides us with that blueprint. And this explains why if you have a problem in your DNA, that problem can get carried through to the message, it can get carried through to the amino acids, and eventually it can get carried into the protein, so the protein doesn't do what it normally does. Now, the last kind of basic concept that I want to talk about is I want to just explain a little bit more about the anatomy of a gene and how a gene actually works in practice. So when we look at a gene, I sort of am going to highlight the attention here. I'm hoping this kind of shows up where I'm circling this top portion of this figure here. And this shows you a little bit about some of the guts of what a particular gene looks like. So a gene is composed of things called exons, which I've highlighted here and I've highlighted here. And these exons, and there's also one here and here. So exons are really important because exons are the portion of a gene that actually goes on to encode what happens in a protein. And these are the portions of a gene that most people think about when we talk about genetics. But it's really important to understand that when you think about a gene, and this will come up later when we talk about complex trait genetics, that there's other portions of a gene, for example, the intron, which are these things that live in between exons, portions of the gene called a promoter, which determine how much of a gene gets expressed in a particular cell or a particular animal, something called a polyacyte, which explains how quickly a gene product is regulated, that can also have mutations in them. And when you have mutations in these portions of a gene, it can be they can be really important in the development of a disease, but it's really hard to explain, at least at this point, how some of mutations that we see in those portions of a gene work. So it's just important to understand that we have lots of different portions of a gene, some of which are easy for us to explain when we see mutations in them, and some of which are harder. So that's really what I want to stop as far as definitions are concerned. We'll come back to some of these definitions as we work our way, as we get through the presentation, and talk about what is a Mendelian trait, a complex trait, and how we're trying to kind of work through solving some of these problems. Okay, so I'm going to go on and talk a little bit, first of all, about Mendelian traits in dogs, so that we all understand what they are, and this is really where the vast majority of the research in canine genetics has occurred, I would say, over the past 10 or 15 years. So let's start off and let's talk about what is a Mendelian trait. So there was I'm just going to change my pointer here. So go back to this. So there was Mendelian traits are named for this Austrian monk who lived in about the mid-1800s, and his name was Gregor Mendel. And many of us may remember his name from basic biology in high school and college. And he spent a lot of his time working with peas. And Gregor Mendel showed us some really important things. And what he did is he studied things like, for example, the color of a pea. So is the pea yellow or is the pea green? The shape of the pea, is the pea round or is the pea wrinkled? The color of the flower, the shape of the flower. And what he showed through his work, through different types of breeding mechanisms, is that these different aspects of a pea, for example, whether it's yellow or whether it's green or whether it's wrinkled or whether it's round, are actually controlled discretely. And the fact that they're controlled discretely ultimately led future scientists who came to discover his work to sort of understand something about the concept of a gene. For example, that there's one gene that controls the color of yellow versus green, and a totally separate gene that determines the color of round versus wrinkled, and a different gene that determines the color of a particular flower. So these are the things that he taught us, and this is why we named some of the traits for him. And he also showed us there can be different ways in which these genes are inherited, and led us to terms of things like dominant and recessive patterns of genes. So just to kind of summarize a little bit about what we mean when we think about a Mendelian trait, is these are inherited traits that are controlled by one gene, right? So for example, again, is the P round or is the P wrinkled? And there are lots of diseases, many diseases that occur in dogs and humans that are inherited in exactly the same way, where there's one gene that causes one disease. And many of these Mendelian traits, they typically follow what we call classical inheritance patterns. And these patterns have names. They're called autosomal dominant, autosomal recessive, or X-linked. And essentially what this means is, do you need one copy of the mutation to develop the problem? Do you need two copies of the mutation to develop the problem, right? That's autosomal dominant where it's 1, autosomal recessive where it's 2, or is it X-linked where it has to do with the X chromosome where there are some differences in males and females? And a lot of research has gone in to trying to identify and study many of these Mendelian traits in dogs, humans, cats, species, all sorts of species. And the reality is that Mendelian traits constitute the vast majority of the diseases that we currently understand, the genetic diseases that we currently understand in veterinary medicine. And so when people when people talk about diseases that are well explained, the vast majority of the time they're talking about these Mendelian traits. Now, there's a great website out there that's put together by the University of Sydney. We call it OMIA, or Online Mendelian Inheritance in Animals. And you can see I've sort of screen captured it here on the right. It's a little bit hard to see in this presentation, but within OMIA, there's been documented over 319 Mendelian traits in dogs where the genes and the mutations are well known. And this is sort of an ever expanding list. People publish mutations almost weekly, if not monthly. You know, I've contributed to this list of mutations, many of my other colleagues at the University of Minnesota, tons of researchers across the canine genetics community that are constantly finding mutations that cause these Mendelian traits. And if you now, the thing that I really want to say about Mendelian traits, right, and this is something that comes from my training as a veterinarian, is that the vast majority of the diseases that we understand the genetics of today for these canine Mendelian traits are either very uncommon when you look at diseases that occur across the canine that are really important in canine medicine, and or they're just very breed specific, right? So they may breed problems, for example, that affect a kidney problem that's specific to poodles, or an eye problem that's specific to collies. And so even though it's fantastic that we now understand and can treat some of the diseases, we can work on removing them from the breeding pool. From a clinical veterinary perspective, many of these diseases that we see are just really not all that common that we see in the clinic. So they're not the common diseases like cancers, autoimmune diseases, many types of urinary stones, lots of different things that cause more common problems in canine medicine. For those diseases, they're just not Mendelian traits. They're just typically not caused by a single point mutation. And so they're much harder to discover. Now, I wanted to sort of take a minute and explain to you, give you some examples of canine Mendelian traits that we do understand, and explain that even for Mendelian traits, there can be complexities or wrinkles that we need to think about when we try to understand, when we think about these Mendelian traits and try to understand them. So an example that I picked, which is one that I like to talk about a lot, is a skin disease, which is called Ichthyosis, which is a disease it happens. I'm not going to sort of say it's common, but it does happen with some degree of regularity in golden retrievers. And Ichthyosis, it's a skin condition where you can sort of see there's a screenshot here on the left of the skin or the hair coat of a golden retriever. And it leads to essentially things that look like dandruff, right? It's these sort of scaly, flaky areas of the skin that will persist throughout the life of a dog that has this. Researchers have worked really hard to identify the mutation for this gene in golden retrievers. It's an autosomal recessive mutation, meaning that you need two copies of the gene in order to have the disease, that you can have unaffected parents that will be carriers of the disease. And depending upon the breeding, in about 25% of the cases, they'll lead to an affected offspring. There's a genetic test for this because the gene has been identified. The mutation occurs in a gene called patatane-like phospholipase domain-containing protein 1, or it's abbreviated PNPLA1. The name of the protein really doesn't matter, but it's been shown that it impairs the development of a particular type of skin cell, and that loss of this skin cell will lead to this generalized flaky skin that we see in golden retrievers. And as I mentioned, the particular mutation in the gene is known, and the mutation in the gene causes something called a premature stop codon. So essentially, there's an error in the genetic code that tells the machinery that makes the protein to stop making the protein 74 amino acids too early, and leads to the formation of a defective protein. And that's what ultimately causes the disease in golden retrievers. This is just sort of a little bit more of a detailed example for you of exactly what a premature stop codon is. And I'm gonna, oops, sorry, I hit the wrong button here. Let me just stop the subtitling. I'm gonna just get the pen here. But what this premature stop codon does is, if you look here at a normal DNA molecule, it's these sequences here where you see CAC, 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 and it leads in the amino acids to produce these three amino acids, GLN, 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 which stands for glutamine, glutamine, glutamine. Now, if you insert a premature stop codon, what you see is at this location, instead of a C, which is what we have in the normal protein, it changes to a T. And the fact that it's now a TAG instead of a CAG, that ultimately tells the protein making machinery to stop producing the protein, and that leads to the premature truncation of the protein. And in this golden retriever's case, will ultimately start by stop production of the protein and lead to the flaky skin that we call ichthyosis. So that's an example of a Mendelian trait. You have one mutation leading to a truncated protein causing one disease in golden retrievers. Okay, but now I mentioned that even Mendelian traits have their own complexity. And this sort of starts to get into some of the challenges that we see as geneticists. So the reality is, if you look at the disease ichthyosis, it actually happens across not just golden retrievers, but across many breeds, right? So this breed here, this little guy is a Norfolk Terrier, this guy is a Jack Russell, and this lady here is a Cavaliers King Charles Spaniel. But if you look, and people have worked out the genetics of ichthyosis across all of these breeds, and lo and behold, what we find is that the mutations that cause ichthyosis aren't in that gene PTPLN-A1, but rather they are in different genes. So this gene called KIRK10 in Norfolk Terriers, TGM1 in Jack Russells, and FAM83H in Cavalier King Charles Spaniels. So this leads to one of the kind of common issues that we see in canine genetics, is just because you find a mutation that causes a disease in one breed, it doesn't mean it's going to cause the same disease in another breed. And it might, to some extent. It depends upon how related those breeds are, what their shared genetic history is. But many times we find that it's a totally different gene, and it leads to some challenges for us, even in trying to understand simple genetic traits across breeds. Another really good example of some complexities, even within Mendelian traits, occurs when we think about this disease called Leonberger Polyneuropathy. And I'm just going to pause up front and say that all of this work has actually come out of the University of Minnesota by one of my colleagues, Jim Mickelson and Katie Miner. Jim recently just retired. He's a really, really well-known geneticist in the canine community, and Katie's worked with him in the lab for years. Now, the Leonberger folks have wanted to sort of make some headway on this disease, which affects their breed. When we say a polyneuropathy, this is a disease that affects the neurons or the muscles of the dog, and it leads to all sorts of problems. So it leads to something called laryngeal paralysis, which is where the vocal cords don't work well. It leads to atrophy or weakening of the muscles. It leads to challenges in the dog's gait or their ability to walk. And all of these things occur within a single breed, right? So just this breed called Leonbergers. And I'm going to highlight a couple of aspects of this slide here for you that I want to point out. So what Jim and Katie and their colleagues have determined over the course of many, many years of studying the diseases is that what we call Leonberger polyneuropathy is actually, so far, at least 3 separate diseases, which they call LPN1, LPN2, and LPN3. So even within a breed, very, very similar clinical presentations, they found mutations in 3 totally different genes that leads to a similar clinical outcome. And so that just goes to show you that even if you try to think about understanding, what is a simple Mendelian trait within a breed, breeds can accumulate diseases that look the same, but when you interrogate them genetically, are actually really, really different diseases. The other thing that I just want to point out here about this slide that Jim and his colleagues have identified is that something called penetrance. So what penetrance means is if you have the mutation, are you going to get the disease? And for 2 of these diseases, LPN1 and LPN3, the vast majority of the time that you have the mutation, you're going to get the disease. But this other disease, LPN2, the penetrance is actually lower. So it means that in most of the cases, so 70% of the cases where you have the gene, you're going to get the disease, but not all of the cases, right? So not every single time does the mutation cause the disease. And this leads to, again, complexities, even within a Mendelian trait, where we really understand the inheritance pattern. So just some takeaways for you for these Mendelian traits. To summarize, Mendelian traits are usually caused by single small mutations in the genome. They're typically easy to explain based upon the location within a gene, right? So that location in the protein in golden retrievers that caused a particular stop codon. So it's easy to explain. We can sort of document what it does. These diseases constitute the vast majority of diseases that have been explained in dogs. Oftentimes, these diseases are rare, and we don't see them super commonly in the clinic. But still, even for these Mendelian traits, there are complexities. So the same disease does not mean the same mutation in a different breed. The same disease can have different mutations within a breed, and not every mutation is fully penetrant, meaning that even if you have the mutation, you don't always go on to develop the disease. So let's switch gears now and talk a little bit about complex traits and what they are, right? Now that we've talked about the diseases, the more simple ones, which we call Mendelian traits. So let's talk a little bit about complex traits. Oops, sorry, wrong way. So complex traits, what do we mean by this? So complex traits occur when diseases can be inherited. But again, getting back to that concept of heritability that I talked about in the beginning, is less than 100%. So the genes contribute to the development of the disease, but never tell us the whole story. And it's possible that things like gene-environment interactions may be important, right? And again, this gets, I mentioned it earlier, the whole nature versus nurture thing, or what kinds of environmental exposures, toxins, chemicals, sun that you're exposed to can also contribute to the development of disease for which there's an underlying genetic predisposition. Another thing about complex traits is that they're typically derived from multiple genes. So whereas with Mendelian traits, it's one gene causing one disease, when we have complex traits, it can be many genes that kind of contribute together to lead to the development of the disease. And because of this, complex traits typically do not follow classical inheritance patterns. So with a Mendelian trait, for example, you could imagine that if you took an affected dog and 2 affected dogs and you bred them together, you're highly likely to get almost all affected offspring. But in a complex trait, you could potentially take 2 affected dogs, breed them together, and because of the different segregation patterns of individual genes, you might not get the outcome you expect. So they don't necessarily follow classical inheritance pattern. It can lead to, for example, diseases skipping a generation, diseases appearing randomly within a pedigree after breedings and breedings where the disease has never shown up. It can just show up randomly like that because all of a sudden the right combination of mutations in multiple genes will show up in an offspring or a combination of offspring. A couple other things about complex traits that I want to point out is that complex traits can also lead to what we call multiple phenotypes. So think about something like height, right? People can be any height. You can be 5'2", you can be 5'10", you could be 6'4". So there's a whole range of different things that can happen. And when you have these multiple phenotypes, we call them a quantitative trait, they're typically defined by multiple genes, again, that don't follow classical inheritance patterns. And then finally, the thing that I want to say about complex traits is that oftentimes mutations that lead to complex traits occur in regions of the genome that are hard to explain. So remember, back when we talked about some of the definitions, we talked about things like exons and introns and promoters. And many times mutations that explain complex traits happen in these regions outside of the exons, where it's really, really hard to explain exactly what that mutation does. And therefore, it's hard to necessarily say, oh, for sure, that this particular mutation causes the disease. So it leads to a lot of head scratching and really creates a ton of work, but a lot of interesting challenges and problems for us to think about as geneticists who want to study complex traits. When we think about complex traits, there's a couple of kinds of complex traits. I mentioned this a little bit on the last slide, but we think about things that are called discrete traits, and we think about quantitative traits. Quantitative traits, I kind of measure already. They're things that we can put a number to. So things like height, blood pressure, hip conformation, weight. And there are things that are discrete, which are more like you have it or you don't. So these are, for example, the disease that I study, Addison's disease, you have it or you don't. Type 1 diabetes, you have it or you don't. Many types of cancer that are genetic, again, you have it or you don't. So different types of complex traits, and when we think about understanding these quantitative versus discrete traits, we have to think about them differently as geneticists when we build models to try to understand these diseases. Now, the reality is that we don't know a ton about complex traits in dogs or humans, really, for that matter. But there's a couple of important complex traits that we really do understand pretty well. And I wanted to take a little bit of time to explain our understanding of these complex traits so that you can get an appreciation for how hard it is to necessarily dissect these, and then to explain 100% how genes explain these traits or these diseases, really just to understand what we're up against and what we tackle as geneticists. And one of the things, what I'd like to start with is sort of our understanding of body size in dogs, because this is something that's been worked on quite a bit by many, many folks. A lot of this work has come out of a fantastic geneticist. Her name's Elaine Ostrander. She works at the NIH, and she's done a lot of work to try to understand the genetics that underlie body size in dogs. And when they first discovered these mutations, it actually made the cover of Science here on the right. This was actually back in 2007. You see here a picture of a Great Dane and a Chihuahua. And what they did in their research is they identified two key genes that are largely responsible for the vast majority of the size difference that we see in dogs, right? So already we're in complex trait territory because we're dealing with height or body weight, right, which is a quantitative trait. And we're dealing with at least two genes, this gene called IGF-1 and IGF-1R, that contribute to the vast majority of the size differences between these dogs. Now, the other point which I just want to make here, right, about body size, and this is something that's unique to dogs, we'll talk about it later, is a lot of the difference in this body size is driven by something we as geneticists call artificial selection. And this is essentially breeders who are people, whoever, who are breeding dogs to look a certain way, right? So Great Danes are bred to be big. Chihuahuas are bred to be small. So the pressure, the selective pressure that we put on dogs as humans led to these sort of two genes segregating to cause these these big differences in the sizes of dogs. Now, since this initial paper came out, and this is where we get into even more of the complexity, researchers have discovered an additional 5 genetic variants that contribute to the body size in dogs. And these are variants in genes called GHR1, HMGA2, SMAD2, and STC2. what the genes do are not really important. But if you take the 2 genes we talked about before, plus these 5 additional genes, this figure here on the right explains a little bit about how these 5 genes come together to allow us to figure out what the body size of a particular dog is going to be. And I'm just going to take a minute and explain this figure to you guys a little bit so you can see how these 7 genes or these 7 mutations come together. So what you see here in this figure on the right is you see these 7 genes or these 6 genes and 7 mutations on the bottom, IGF-1, GHR1, SMAD2, et cetera. And on the y-axis, you see the weight for a particular dog breed, right? So we have small dog breeds here at the bottom, and we have really, really large dog breeds here at the top. The weight just goes up. And within these boxes, you see some yellow and you see some red. And essentially what it means if you see a red dot is that dogs that have what you see is that typically when we see a red dot, animals will have 2 copies of the gene in question. And where you see the yellow dot, animals will have essentially 0 copies of the, not of the gene, of the mutation, of the variant in question. And so when you look at all of these genes together, you can see that these small dogs at the bottom here, across all of these genes, are really, really red, which means that they have 2 copies of the variants of all of these genes that together are contributing to having a small dog. And as you go up and up and up in body weight, you can see many more of the animals have 0 copies of these variants in all of these genes. So having 0 copies of these variants is what makes a big dog, and having 2 copies of all of these genes is what makes a small dog. And you can see there's a bit of a gradation in the frequency of these mutations across all of these breeds as you go across the different genes. So the whole point here is that all of these genes really come together to determine the body size in dogs. But even with that, right, even with that, we probably can explain only half of the body size difference among breeds, meaning that there's still work to go beyond just these 7 genes to understand body size in dogs. Another example that I want to point out to you guys, or I want to talk a little bit about, because the reality is that even though we've done a great job in studying body size, very, very few clinical diseases that we see in our patients that are complex have actually truly been dissected. And there's one exception to that, or 1 or 2 exceptions to that. And probably one of the best examples that I want to go through with you guys here is this disease called dermatomyositis in Collies and Shelties. So dermatomyositis is a disease, it's actually a vascular disease, so a disease of the blood vessels that it occurs in Collies and Shelties primarily. And it leads to essentially these red blistering pustules and this area of no hair or alopecia on the limbs and the feet and the tip of the tail and the nose within these breeds. So it's a pretty severe and disfiguring disease for Collies and Shelties. And I'll just sort of say that all of this work has been done by a fantastic geneticist, one of my colleagues. Her name's Leanne Clark out of Clemson. And she pioneered this work with some of her grad students. And what she did through the years that she spent studying this disease is she found that 3 genes interact together to predispose to the development of this disease within Collies and Shelties. And these genes are in one called the major histocompatibility complex, a gene called PAN2, and a gene called MAP3K7CL. What these genes do are not really important. And in fact, the exact role of at least 2 of these genes, we don't really even know. So we've identified markers in these genes, but we don't really know the roles of these genes in the body. And getting back to one of the points that I mentioned about complex traits before, is that the location of the mutations in all of these genes doesn't actually alter the protein structure. So it becomes really, really hard for us to explain what these genes actually do. And so, but even though we can explain the disease by understanding these mutations, we don't really fully understand it because we don't understand the genes, nor do we understand exactly what these mutations do in the future within the protein. The other thing that I want to sort of point out here that is that that what's really, really interesting about this particular mutation, as I mentioned, you need these three genes. Here we've labeled them as A, B, and C. So you see them as the purple bar, the gray bar, and the orange bar. And how long the bar is has to do with how common the mutation is within a population of dogs. And so what you can see in, for example, the Collies and the Shelties that I've highlighted here, there's pretty high frequencies of the purple gene, the gray gene, and the orange gene. And together, the frequency of all of these genes in the Collies and the Shelties is much, much higher than what we see in other breeds. And if you take a look, for example, at like a Boston Terrier, so a Boston Terrier has a really high frequency of the C gene, but it has a really low, none of the A gene and a very low frequency of the B gene, so that Boston Terriers don't get the disease. And this is another thing that we kind of see within complex traits, is that you need the combination of multiple genes to cause the disease. And even though you see some of these, quote, mutations in other breeds, it's only when they come together in a particular way that an animal develops the disease. And as Dr. Clark has shown in her work, the only instances in which these mutations come together are within Collies and Shelties and not other breeds. And that explains why we see the disease in this breed and not others. So just some takeaways for complex traits. So complex traits, or many variants, contribute to the development of the trait. Genes are not going to explain everything, so we need to think about things like environmental effect, the whole nature versus nurture. Common variants that we see may only have really, really small effects, and important variants can live within unexpected genes based upon their currently understood function. Now, before I kind of get on, and we're going to wrap up here in just a few minutes, but a lot of the questions that I got were around genetic testing, right? And I want to just point out here that as we start to understand more and more about complex traits and understanding that, okay, you need these 3 or 4 genes in these proportions to actually cause the disease. One thing which I'll say up front is, again, we have very few genetic tests for complex traits, but you're unlikely to ever get a black and white answer. So it's never going to be, oh, if my dog has this, it's going to definitely get to the disease. We need to think more about disease risk. So if my dog has this combination of mutations, it's going to be at higher risk of developing the disease, but not necessarily 100% get the disease. So that's really what I want to say for now about complex traits. And lastly, as we kind of finish up here, I'm just going to go through quickly some of the things that researchers are trying to do to sort of tackle some of these issues around complex traits. And this gets back to when, after the publication of the human genome, back in the early 2000s, researchers started to bring up this question of what I like to call, or I like to bring up, called the missing heritability. So it's like, why, when we understand something about so much about the structure of genes, about the nature of the genome, about the basis in the genome, can we still not explain all of the causes of genetic diseases? And this really applies, right? And this is what I mentioned earlier, to so many important canine inherited diseases: Addison's disease, diabetes, cancer, hypothyroidism, lipidemia, pancreatitis, heart diseases, dilated cardiomyopathy, Wobbler's disease. All of these unexplained canine inherited diseases that we see so commonly in the clinic, these are complex traits. And the reason why we have such a hard time understanding them is because a lot of what we focused on to date are these Mendelian traits that just don't explain a lot of these more common diseases that we see in animals. And explaining the more complex traits takes a lot of time and effort and work. I'm going to kind of skip over some of these areas around missing heritability, but what it really boils down to is that a good chunk of the reason is why we haven't been able to explain a lot of these medically important diseases in dogs is because they're complex traits, and it takes a lot more effort to try to understand them. But there's a lot of things that we as geneticists are doing. So for example, we're developing new tools. We're developing things like whole genome sequencing, better genetic markers, improved reference genomes, and also like new approaches. So things like environmental awareness, better ways of understanding animal's clinical picture, methods for doing things like mapping traits that are fixed in breeds, really to try to get a better handle on mapping these complex traits and understanding them so that breeders can have genetic tests, so that veterinarians can sort of use them to understand disease risk, and fundamentally so that we can improve outcomes in animal health, which is the whole goal of what we're trying to do here. So I'm going to stop there just in the interest of time, and I'd like to kind of turn it over and we'll answer some of the questions at this point.
Yeah, thank you so much, Dr. Friedenberg. A great presentation. It was really helpful to have something so complex broken down step by step. So let's jump into some of these questions that we received previous to tonight and the ones that we got while you were speaking. So can you share a little bit more about some of the common diseases dogs get? I know you referenced these like cancer, IBD, IBDD, DCM, etc. And if there is research that has shown any heritable links there.
Yeah, so definitely there's a lot of research for many of these diseases, like the ones that I put up there, like IBDD for sure. There's been a lot of work looking at that disease. But again, many of these diseases are in fact heritable. And part of the reason we know they're heritable is because they tend to aggregate within a breed, right? So for example, like a heart disease, like dilated cardiomyopathy, we tend to see that in Doberman Pinschers. We tend to see that in Great Danes. We tend to see that in Irish Wolfhounds. So we know that those diseases are heritable. The challenge with them is that they're just heritable, but they're also complex, right? So it takes a lot more work to kind of understand the genetics of those diseases. But there's certainly a lot of research out there on many of those diseases. I think we just don't have the complete genetic picture or the genetic story on many of them at this point. But lots of researchers out there like myself trying to explain this and trying to understand these diseases.
Sure. So how can someone, as a person trying to make sure that they're getting the healthiest dog from a breeder, for example, know what to ask the breeder to confirm that best practices are being used?
Yeah, so if you're working with a breeder, you're probably most interested in, you're obviously interested in a particular breed. And I think the thing that I would always recommend up front is to kind of do your homework, right? So to look at what are the common genetic diseases that occur in your breed, to investigate whether or not there's a genetic test out there for those diseases. And there are, for example, you could look at that online Mendelian inheritance in animals. You could look at a lot of genetic testing companies out there that will list mutations that they test for. And then you can ask your breeder, does your breeder test for this common disease within the breed? Now, just keep in mind that there are a lot of common diseases that occur in breeds for which there are not genetic tests, right? So for example, a disease that I study, Addison's disease in Portuguese water dogs and Sandra poodles, there's no genetic test for that. So a breeder can't say that, oh, hey, this dog is free of Addison's disease, or this dog is definitely going to not develop Addison's disease. There's just a way of knowing. But I think if you're interested in a particular breed, do your research, try to find out what genetic tests are there, ask the breeder if they're doing those tests, and kind of go from there. I think another question that often comes up within breeds is, I think, you want to sort of get a sense for how breeders pick the population of dogs that they breed to try to make sure that they're breeding as unrelated animals as possible, even within a breed. So that's another thing.
Okay. So the next question is about mixed breeds, actually. So are mixed breeds less prone to genetic diseases? And if deciding on a purebred, is there any way to minimize the risk of getting a pet with a genetic disease? So I think that person's kind of weighing between the two.
Yeah, so for sure. So I think mixed breeds definitely are less predisposed to developing genetic diseases. And that's particularly because a lot of canine genetic diseases tend to congregate within a breed. And so if you try to mix the breed, right, you take a breed that's really, really highly predisposed to like dilated cardiomyopathy, and you take a breed that's not highly predisposed to dilated cardiomyopathy, you breed those dogs together, there's just going to be inherent less genetic risk in developing that disease. Now, that doesn't mean that a mixed breed dog can't develop a genetic disease, but they're definitely at lower likelihood of developing those diseases. So, from that perspective, it tends to be safer to avoid genetic diseases to get a mixed breed dog, but you can't certainly always avoid it.
Sure. I know we've talked a little bit about tests. So this is a specific question about a test. Are results from DNA tests like EMBARK actually helpful to veterinarians?
So, I think EMBARK testing is great. I think they provide a summary of whether or not your animal is at risk for many, many different genetic diseases. Sometimes they provide, for example, the results of a test like, we talked about the ichthyosis in golden retriever as well. It doesn't really matter if your dog has that mutation, if it's not a golden retriever, but you're still going to kind of get that result on the EMBARK test. But as a veterinarian, I can't say that any of those tests have really proved very useful for me. Have really proved very useful. I think at this point, the genetic tests really help breeders make sure that they avoid certain combinations or certain animals that may be at high risk of developing, important inherited diseases within those breeds. But again, those typically apply mostly to these Mendelian or these single point mutations.
Sure. So maybe you just answered this one, but there might be a little more to add. How should dog owners interpret any genetic testing that they get from common breed diseases for their dogs? And how should owners then use that information to direct healthcare choices throughout their pet's life?
Yeah, so, I think it depends, right? I mean, I think, ultimately from so for example, we're not at the stage, right, for example, where we can take a dog's genome, sequence its genome, and sort of predict exactly like what kinds of health problems they have. I think with the way genetics work, breeders can kind of use genetic tests as sort of a forward-looking thing, right, to understand who do I breed together. But as an owner, once you have a dog, and let's say you have a golden retriever and it sort of one day presents with this flaky, scaly skin, you have a genetic test run on it and it turns out, oh, it does have the mutation for ichthyosis in this breed. Well, you can certainly tell that to the dermatologist, right, that my dog has the mutation for ichthyosis, but I would bet your dermatologist is going to know that, oh, this is ichthyosis already and doesn't really need the mutation to inform him or her, how to treat your dog. So I'm not 100, I don't really think we're at the stage yet where genetics is informing too much around veterinary healthcare, but I do think that that's rapidly changing, right? So for example, as we start to understand, for example, more of these complex traits, and we might be able to say, oh, well, based upon the combination of genes that your dog has a 50% chance of developing Addison's disease. That could, number one, help an owner to make sure they're more aware of what to look for if their dog does develop Addison's disease. Or number two, it could make us figure out, well, what steps can we take to reduce the risk of actually developing the disease? So I think kind of from a clinical perspective, we're not quite there yet as far as genetics are concerned, but I would say that's stuff that's going to be coming down the pipe over the next 10 or 20 years.
Sure. So the next question is asking if you or your peers here at the university plan to utilize the genetic database being compiled by the NIH's dog aging project.
Yeah, for sure. So I think, there's a project out there which is funded by the NIH. It's something called the Dog Aging Project. And the Dog Aging Project it's very well funded. There's going to be doing a lot of genetic research, collecting a lot of environmental data, understanding many aspects of the dog's health, and try to understand, what's the impact on aging. The Aging Project is going to be pulling together a lot of publicly available resources. And I have no doubt, because those resources will be such high quality that myself, other researchers will definitely make use of those as we study our own diseases or the disease we're interested in for a whole host of reasons. So
sure.
For sure.
Yeah. Okay, a question that came in is, are genetic diseases less common in wolves or coyotes than in domesticated canines?
It's a great question. I'm not quite sure, I know the answer to it because I don't study wolves or coyotes. But the one thing which I sort of will say is that my hypothesis or my guess would be that they're less common. And part of the reason why I think genetic diseases can be so common in dogs is because we breed them to all look alike within a breed, right? If you're talking about a breed specific dog, is because we breed them to be the same. So we put this pressure on a dog breed to look the same. And when we put that pressure on a dog breed, we sort of limit or narrow the gene pool, and that can lead to a congregation of diseases within a breed. And that's something that, wolves or coyotes that sort of don't have humans orchestrating who breeds to who might be less likely to experience. So that would just be my guess.
Sure.
I think, the other thing is that, if you have a sort of a highly unfit animal that lives in the wild, that animal is a lot less likely to survive and reproduce than, for example, a pet that might be sort of, taken care for with like medical care. So.
Right. I think the next couple of questions definitely played off some of what you talked through in the presentation. So how much is known about the POMC mutation that causes Labradors not to feel full? Is it complex versus not, or heritable, or what can you kind of tell us about that if you know that specific mutation?
Yeah, so I'm definitely aware of this mutation. So there's a mutation that's been shown. It's sort of really interesting in Labradors in a gene called POMC. It stands for pro-opiomelanocortin. And there's some notion that it leads Labradors to sort of to never be satiated, right? So that's kind of why they eat and they eat and they eat. And as to the best of my knowledge, that's not necessary. That's not a complex trait, but where it's because the mutation's kind of clearly been identified. But I just don't know that I know enough about it. But I know it's a pretty common mutation within Labradors. So.
Okay, and this is maybe kind of similar in that, is epilepsy considered a complex trait, is a question?
Yeah, so definitely I would say epilepsy is probably a complex trait. But the other thing I would say about epilepsy is that epilepsy, right, is something that we classify clinically as we see a dog that has seizures. But there may be 100 different genetic reasons why dogs will get epilepsy. And the other thing is epilepsy may not be entirely heritable, right? So for example, let's say you have a dog that was hit by a car and sustained some kind of trauma to its head. So that dog can develop epilepsy because it develops some trauma to its head, and there's a region of its brain that's sort of no longer normal and leads to a focus of seizures. So that's a totally non-genetic reason for developing epilepsy. But if there are genetic reasons for developing epilepsy, that disease may not be 100% heritable. So the genes may not be the only thing that lead to the development of the disease. And I would argue that it's probably going to be, even if you did understand this mutation, they would likely be very, very different across breeds because there are so many different common pathways that can lead to the development of seizures.
Okay. Probably have time for one more question. So what makes dogs have such morphological variation? Cats look much more similar to each other.
And yeah, that's a great question, and I think the answer to that question is really us. Like human beings are what make dogs have such crazy morphological features because we've bred them to look a certain way, right? So for example, we talked about in this presentation, we talked about height. We've bred Great Danes to be really big. We've bred Chihuahuas to be really small. There are other mutations that for certain morphologic features that are also known, right? So for example, we know that the mutation in the gene that causes a screw tail in like pugs and English bulldog. We know mutations that cause like longer furnishings or certain different types of whiskers. We know mutations that cause different coat colors, right? But the reason why these dogs look so different, especially within a breed, is because we've put pressure on them to look a particular way by essentially making those mutations congregate at a really high frequency within a particular breed. So, that's essentially, it's all because of us.
We play the role in that.
Totally.
All right. Yeah. Well, great. Thank you so much, Dr. Friedenberg, for this great talk. And thanks to our many participants who shared such thoughtful questions in advance and during the presentation. As always, the Veterinary Medical Center is here for you 24/7 to help care for your pet. So please don't hesitate to reach out to us. And if you enjoyed learning about our team's work tonight, we do encourage you to visit our website or reach out to us directly to learn more about our mission to improve the health and well-being of animals and people. And we do hope that you'll consider supporting the Canine Genetics Lab through philanthropy. Gifts certainly play a critical role in expanding our work, and we thank everyone joining us tonight who does support initiatives across the College of Veterinary Medicine. And next month, we look forward to welcoming Dr. Anna Brutlag, who will join us to discuss the work Pet Poison is doing to help owners identify potentially toxic items and help keep pets healthy. So thank you again, everyone who attended tonight, and we look forward to seeing you next month.
Thank you, everyone.